guys. I know that there were college applications that have been due, and I know it is the end of the quarter, although you realize for us it's just a snapshot for you because we have a rolling grade. I know that you all are interested in making sure your grades were good for AP Bio. So I know it's been pretty intense. All right. So that being said, I was looking over things and looking at grades and thinking that there's so many points in the practice and there's almost none in the summative, and that seemed it didn't seem well balanced to me. So I've made an executive decision, and what I'm going to offer you, and I hope that you see this as a positive thing, is that I want to reclassify. Let me explain exactly what that's going to mean. Your ed puzzles on where I was showing you the potato lab, and the ed puzzle where I was showing you fusion through dialysis tubing. Two of them. There are two of them. You already did them. You already did them. They're already part of the practice that you did for a 2.5. And I didn't grade them for accuracy. I just gave you a 10 out of 10 if you tried. What I'm going to do is I'm going to look at your actual percentage that you got, and I'm going to put them in as summative grades. So before you panic, if you got in the 90s or higher, I would say that's really going to help you. If you didn't, I'm giving you the opportunity to redo them. I'm giving you the opportunity to redo 2 ed puzzles, which are then your own individual lab work, and will go in as 25-point labs each. So together they'll be worth exactly as much as your muscle CER was, except it's all you. And you have until Saturday to redo them for perfect grades. I've just put all the grades that you did get on them, I put into your Google Classroom grade sheet. I just sent them out in the last hour. So you can go and look. Some of you did well on both. You don't have to do a thing. Some of you may want to redo it again. I just refreshed it so you can just go in there, really concentrate, really get them right. Go for it. Okay? These are appropriate to be labs because everything that I asked you in those 2 ed puzzles are really appropriate to what I'm going to talk about today and what you're going to need as skills for next week. Because some of you are very seeming like you're really confused still about a lot of things in terms of graphing skills, in terms of how to write a title, in terms of picking your IVs and your DVs. Nobody understands sample size. None of you got that right. So I think it's worth going back over, and it's worth giving you a chance to have that extra summative grade, which can go into quarter 1. And then I think for most of you, that should be a bump if you go in there and you concentrate. And it's not asking for a lot. So I want to see how you feel about this. How many of you are thumbs up about this? My mic is on. Yes. Okay. All right. Yes. Good. All right. Ella, did somebody have a question? Please don't hesitate to ask questions. This is a good time. Yes? So again, you can go and look at your grades. They're in Classroom, your Google Classroom. They're in there now. For the ed puzzle, here, I'm going to write it down so you guys know what I'm talking about. Okay. The ed puzzle on diffusion and the ed puzzle on the potato lab. And both of them are Mrs. Murdoch. Yes, anyone, Lily. Yes, you can do it. If you have a 98 and you want the 100, go for it. Okay, it's fine. It's difficult for me to check. That's one reason I'm willing to do this. I don't have to sit and read anything that you guys have written. I just need to see what your percentage is on ed puzzle. So that's not really onerous for me, okay? But I think it's worth doing, and I like the questions. I just looked at them again this morning. I like the questions I was asking you. They go right with what I'm going to talk about today. So let's just do it, okay? So if you got 100 on one and a 50 on the other, redo the other one, okay? If you got 95 on both, leave it alone, go on to other things. It's just up to you. Okay. If you didn't do it at all, definitely do it. But don't start that until we've had our talk here, because everything I'm going to talk about with you right now is going to help you do hundreds on those. Okay. And again, hundreds on the big lactase enzyme we'll do next week. Also going to help with that. So not wasting your time here. Okay. All right. So hopefully you've gotten to look at your muscle rubric that I emailed you, right? If you didn't at this point, let's just go forward, because there are things that I just like to speak about, and I just want you all on the same page. But so now you know those two items on the board right there, the Ed Puzzle on diffusion, the Ed Puzzle on potato lab. They're both Mrs. Murdoch explains things, right? Where I'm wearing my apron and I'm talking to you and I'm asking questions. Okay, so they're fresh up on. Let me just go show this. To your class period, UR6 period. Okay, so if you look in classwork, there's a bunch of things that are in this 3.1 enzyme structure and function place. And there you go, potato and diffusion Ed Puzzle redo. So if you open that, okay, it explains what I just explained. I'm reclassifying these as summative, 25 points each. If you already did really well, ignore this. If you don't like your original grade, which I just posted on Google Classroom, you can redo them. Doesn't matter what your original grade was. If you had an 88 and you want the 100, go do it. Okay? If you already have grades you're happy with, leave it alone. I thought that might help with things. All right, so what else we're going to do today, we're going to be looking at some graphing skills and ways to help you do better on some of these graph and CER things that I've been letting you do here over the last couple months. So go back here, see what you're seeing. Yeah, all right, so you saw that. So now, okay, so first of all, lots of you, and this is still going on because I just went through all your mystery sucrose molarity assignments, and I give you grades for those too. Those are just practice. Those were like those I should do for 2.5 or something. So it's not really high stake. So if you struggled on that, that's way less serious than if you struggled on the muscle CER that you all turned in that was the grade, right? But one thing that seems to be true for everybody is that you are having just really the main thing I want to talk about is how to write a title. Okay, a title for a graph. And I'm not just giving you all these directions and asking for all these things because I'm a fastidious teacher who wants too much detail. This is college level. This isn't just college level. This is professional level. This is if you're going to present a graph to anybody. It doesn't matter if it's science or if you're, in the corporate world, or if you're running a business, whatever it is. If you're presenting a graph to people, that graph should be able to stand on its own. You shouldn't have to go and read the procedure of the lab to figure out what's going on in the graph. It should be obvious from the graph title that it should be obvious what happened to get that data. It should be obvious. So some of the things you need in the graph title, right? IV and DV need to be correctly identified. Correct and related correctly, right? So, for example, I got some really crazy titles on some of those sucrose graphs where you were telling me things like the color of the sucrose solution affected the mass change. Literally, I had people telling me that. Or I had the initial mass of the bag is the final mass of the bag, which is way out there and shows that you really didn't understand the experiment at all. or you know, I had just not even anything. Percent mass change of dialysis bags. No relationship explained at all, right? So number one. Another thing very important in a title is if it's, you need the sample size. And a lot of you are extremely confused about what sample size means. So let me just go over it quickly. Definition. Sample size is the number of test subjects receiving each treatment. Okay, so let's explain that a little bit, because I think a lot of people and a lot of adults, I think, would be confused by this. So don't feel bad if you got confused by this. In the let me just draw a picture. Let's say, for example, that I've got some cups. Let's talk about the potato lab, since I haven't looked at that yet. Hint, hint. And if you fix it before I go look at it tonight and the next night, you might get 100 on it. Let's say that we've got 6 cups, okay? And we have solutions in each one of the 6 cups. And this is 0. There's nothing out here. And this is 0.2 sucrose out there. And this is 0.4. I'm just going to do a few. And then this is 0.6 molar sucrose. And then this is 0.8, right? And then floating in those different solutions, you've got some potato slices. Potatoes in. Okay. And that is my experiment. That's it. That's all. I haven't done anything else in my lab other than what you see right here. And let them soak. I get an initial and a final mass, right? That's the experiment. Okay. So looking at this, first of all, what are my test subjects? What am I using for test subject here? What is that thing there? Potatoes, yes, that's it. Okay. Potato slices are test subjects, right? Okay. So how many times did I subject test subjects to these different treatments in this experiment? Just right here. Yes, that's it. That's it. Okay. So for each treatment, and there's 5 different treatments here. That's okay, Elsa. Don't worry. That's why we're doing it. That's okay. So for each one of these treatments, there's only one test subject right now in this picture being treated, right? So for this situation right here, n equals 1, because I didn't repeat the experiment any number of times. I did the experiment only once, and I have a test subject only being treated with each of the levels only once, right? So a lot of people were doing this. You're confusing sample size with the number of levels of your treatments, right? So you might look at this and think, well, sample size, there's 1, 2, 3, there's 5 cups, so it must be 5. The five cups are each different from each other, right? So in this case, this experiment was only done once. There's only one trial here. That's another way of thinking about it. And the test subjects were there's only one time that the test subjects are being treated with these different things. Oh wait, that was a good question. I missed it. Sorry, let me go back to that. But if you were to repeat it 5 times, right? Even though you have 25 potatoes, the sample size is 5. Yes, yes, right. So like, let me just draw some more pictures for you. What if I did it like this? What if I set up my lab? I'm not going to bore you with lots of drawings here, but what if I had that same situation, same molarities in each cup, right? There's my potato in each cup, same concentrations as the one above them. There we go. And then what if I could do this again, I had another set of beakers here, right? Same levels of sucrose solution. Potatoes are, you know, from the same species potato. Those are, oh, sorry about that. Fingers are getting a little crazy there. But now what's my sample size? This was, yes, there you go. There you go. So the sample size really is how many times you repeated the experiment for the test subjects, right? How many times did you subject test subjects to 0.8 molar sucrose solution? 3. How many times did you subject test subjects to 0.6? 3. There you go. So that's what sample size is. So now if you relate that to, right, so now n equals 3 because you took these test subjects and you repeated their treatments 3 times. Okay? So the sample size is not how many different levels of treatment you have. The sample size is how many times you subjected test subjects to those treatments. I am recording this. I like the way I said that, so you'll be able to look at this later. Okay, good, good. So hopefully that helps clear that up. Now with the muscle lab, what you had to do was you really had to read the description of the experiment carefully. And what it said was, let me just erase this. The muscle lab, in the very last bit of the paragraph question, it actually told you that, like, let me just draw a long island here. I'm just gonna, oops, this could be ugly. All right, so here's Long Island there, okay. And here's the sample size, right? It was kind of like this, wasn't it? And then as you got out towards the Atlantic Ocean out here, it got saltier and saltier. So there was your map, right? So the salinity, and they were numbered, weren't they? This is a good one to have because, yeah. So this is what the paragraph was explained, if you were careful and you really listened, that at each one of those test sites, let me just, this is long. There it is, and the Atlantic is out here. Remind you. There you go. So at each one of these test sites, how many total muscles did they get from one test site? Some of you have hopefully your paper out in front of you and you may remember. I had a lot of different answers for this. Some of you said that, oh, sample size is 8 because there were 8 test sites, right? A lot of you said that. And that again is the same misunderstanding as with the cups I just showed you. The sample size isn't how many different treatments you do. Each one of these test sites is a different treatment because it's a different salinity at each test site. So 8 is not the correct sample size. The sample size is how many muscles did you sample for each site? And the answer is 100 if you read it. They took 100 muscles from there and 100 muscles there and 100 muscles, and they repeated that sampling number for each of these different places where the salinity was different. Right? Then I had some people who misunderstood that and thought that the sample size was 800, which also is not correct, because 800, yes, that's how many total muscles were sampled, but that's all 8 different levels. So that's not how many muscles were treated with each treatment. So the correct N value, which in your graph every time, okay, if you had more, if you know how many samples you had for each treatment, you need to put it in your title as an N equals value. In this case, N equals 100 because they repeated the treatment 100 times. They counted 100 muscles and figured out how many of them had that gene they were looking for at each site. How are you feeling about that definition? Are you feeling better? Give me a thumbs up. I understand sample size. Just give me a thumbs up on that. Yes. Okay, good. Okay, excellent. All right. So now since we've got open, the other thing, oh, great, Jack, I'm glad to hear that. All right. The other thing that I think people got really confused about, and that I was listening as you were doing the essay, one of the other really confusing things is, what's the IV here? You all understood that the frequency of the lap allele was the DV, right? You got that. You understood that. I don't think very many got that wrong. But you were very confused about the fact that I gave you site numbers and then I gave you salinity values, and you weren't sure which one was the IV. So I really want to make sure I make that very clear, right? So let's talk about that for a second. IV and a DV. Independent variable, right? Versus dependent variable. So we'll go back. The dependent variable is the value that depends on the variable scientist is manipulating, right? So the dependent variable is usually what you're measuring as a result of something else that you did to the environment to mess with your test subject, right? So what's the IV? The IV has a couple different ways of being explained. You can say that often it is the manipulated variable is one way you've heard it explained. You could also say that it is a variable scientists are curious about studying, right? If I add that little bit there, let's really look at what the true IV is for this, right? I mean, yes, you had numbers, you had site numbers. And it is true that the site numbers definitely determined, because of their geographic position, they determined what the salinity of those sites were going to be, right? But the true IV here, the thing that is going to drive the frequency of the alleles, is not where they were geographically. The true IV is the salinity. The salinity is what the scientists are really curious about, right? Scientists are not trying to find out whether the geography of a sample is going to affect the salinity. They're already pretty sure. They're not asking that question. They know that the closer they go towards fresh water, right, the closer they go towards fresh water over here, the salinities will be lower. That's why they chose those sites, right? So you don't want to shoot down the sky with the sky here. What they're really curious about, the driving IV here, they are manipulating so that they can see what effect it has on the muscles genes, is salinity. So knowing that is going to lead to the correct title, right? Is there anyone who had any questions about that? Okay, so if we know that, and we know that the salinities did get higher as the sites went out towards the ocean, now let's look at what would be the best way to title that. So let's go to where. Here we go. No. Here it is. Okay, let me just share my screen. I'm going to share my screen. It's going to accordion its way down. There we go. Let's look at this first. Okay. So ideally, because you have salinities, numerical salinities, those salinities needed to be on your x-axis in some way, okay? Or they needed to be reported in some way. The best graph that you guys could have done, truthfully, would have been a scatter plot with a trend line. But I had taught you that one. So if you did a line graph, you had had those, and that was fun. If you did a scatter plot with trend line, that's great too. You did fine if you did either one of those. But look at the title I've got. Look at this. The effect of salinity, parts per thousand, on the frequency of LAP94 alleles found in mussels sampled during a field study in 8 different locations in Long Island Sound and the Atlantic Ocean, and equals 100. Now that's not a huge title, but it hits all the boxes. That's what they're looking for. That's what AP Central is looking for. That's what I'm looking for. That's what they will look for if you ever do graphs in college or as a professional. You need to be giving people the information, right? Now, you don't need to go overboard. You don't need to do the whole lab procedure in your title. That's not what I mean. But look at all of the details that are here. I understand from reading this title now that, oh, they're looking at salt effect on an allele in the mussels. And they sampled, and this was a field study. It was done outdoors. And 8 different places, around Long Island Sound. And, I know where that is. And a nice big sample size. Look, 100 mussels per site. That might make it pretty reliable. Okay, so that's what I'm thinking as the reader. And that really is everything you need. So the other one that some people turned in looked like this, and I really didn't take off that much. The reason why the bar graph is not ideal here is because the salinities are not represented at equal intervals down here. So this skews the data a little bit, right? This curve is a different curve, really, than up here. Just because 10, 15, 18, there's an interval of 3 there, and there's 5 here. So you have numerical salinities that are not well represented correctly on your x-axis with a bar graph. I took off a point if you did that. Okay. How is that feeling? Are people feeling a little better about that? Back to you. Oh, good. Excellent. Excellent. That is what I hope for every time. Okay. All right. So let's make sure I don't leave anything out here. Okay. Sample size. Yes. All right. So which graph to use? We're going to watch Mr. Anderson in just a minute, and he really pulls all this together beautifully. And then with all of this as well, it'll make it easier too if you want to go in and redo those ed puzzles of me doing the potato lab and the diffusion lab. The things that I'm telling you now are really going to help. Well, okay. So let's see. Writing tips. All right, I don't want to go into this too deeply, but I wanted to share with you something that I already put in your, excuse me, in your enzyme classwork. So let me just give you. Here we go. Get a little open. Yes. Look at that. Okay. All right. So hopefully you can see this. Again, I can't see your chats, or maybe I can. Can I? Oh, yes, I can see your chats. So I gave you a copy of this. Half of your AP exam is essays, guys. So this is something that we need to work more on. And I know we've been concentrating more on graphing and just really kind of skipping over the CERs. I'm going to try to do better at that, okay, especially with next week. The lactase lab, I really am going to be looking at everybody's writing ability. So you're going to need to pay attention when I give you these kinds of tips. So I found this. Another teacher had written this. Somebody who's been an AP reader wrote this. So they're looking at your writing the same way I am. So some of the tips that they're giving are, remember that you're trying to show understanding, mastery of introductory bioscience. Okay, so graders are looking. We're looking for answers. We have a list of things, specific terms and definitions that we're looking for. So that's where we're coming from. Always write in complete sentences with your best spelling and grammar. There were so many errors, guys. Spelling errors in your muscle, and that you've got a spell check in there. I know because I've used it. Use the spell check. Use the good grammar. make use of that because it's there. Write something that's going to be, that's not going to be confusing for the reader. Since you have grammar and spelling checks in there, make use of it. Don't do a whole bunch of flowery introduction sentences like diffusion is a really, really fascinating process, and I need to talk with you all about it. You're wasting your wrist muscles writing stuff like that. We don't need to hear it. What we're looking for is that you go right to the meat, right? So no long introductory paragraphs. This is not like an English essay. This is science writing. So straight to the point. Okay, let me go back and. Okay, so if I keep going, I'm not going to spend a huge time. I already downloaded this for you, so you can look at it yourself. All right, organize your answer the same way the question is organized. That was no problem because I had it organized for you and you put it right in. But when you get to the real essays, do the same thing. So label things as A, B, C, or D, so it's easier for the AP readers to find it. AP essays are corrected on a point system. The grader is looking for certain specific statements or answers to award points. So if you're supposed to describe mutualism, first the grader is going to look for the definition, and then they'll also probably look for an example of it, right? All right, always answer the question asked. Okay, there we go. So some of you on the CER part of this, I think the question that was most often missed on everybody's paper was when I asked you to tell me to describe what type of molecule LAP94 is and what its function would be in a muscle cell. This really could have come straight off of that that graphic organizer that I gave you. It could have come straight off of that. I told you that an allele was a gene. You should be able to know what kind of molecule genes are made of. You should be able to know that that's DNA. LAP94 was a bit of a DNA molecule. And if you got that right, the function of DNA, all you had to say was it stores genetic information. Many of you were overthinking it. Many of you were already trying to talk about salinity or this or that or proteins, amino acids. No. Read the question. Just read the question really carefully and answer exactly what it's asking. No more, no less. What type of molecule is it? Okay, so it's allele. Oh, allele is a gene. What's a gene? A gene. They're in the chromosomes, right? DNA. Okay, so DNA is the molecule. And then what else? Or nucleic acid. You could have said that too. I would have accepted that. What's the function of DNA? To store genetic information. Or you could have said it carries the information necessary to make the LAP94 protein, the enzyme. From the biochemistry unit, you should have been able to do that. But again, a lot of you were overthinking things and getting confused, right? So again, I like these essay tests because they really test whether your mind is well organized with the information. And if you're going in, with the graphic organizer ready and with everything in the right categories in your mind already, you'll have a much easier time. You will not get these answers on the internet. Don't even try. You won't. I wrote them. I wrote the questions. I know what I'm looking for. They're not out there on the internet. But don't bother. Okay. Then I wanted to show you my very favorite answer that one of you did, and I don't know who it was, but it was brilliant, and I just want to show it to you really quickly. Okay, there. I'll show you that. Where is it? I think it's this one. Okay, hopefully you can see that. A little bit small. No, let's do it. All right, we'll just do it here. Very small. All right. Bigger. I don't know who wrote this, but it was very well written. That last question, which I knew would confuse a lot of you, is why exactly Why do you think mussels living out in the Atlantic have more alleles than the ones in Long Island? This person, whoever they were, answered it perfectly. The Atlantic Ocean has a significantly higher salinity level than the bodies of water near Long Island Sound. Short sentence opens up what's in the environment that we're curious about. Over time, natural selection in areas of high salinity caused an increase in mussels with the LAP94 gene due to increasing their tolerance of the salt water. You didn't have to explain natural selection, but if you mentioned it and you showed understanding, that's great. Why this happens is because LAP94 enzymes remove the N-terminal amino acids, which creates a higher concentration of amino acids in the cytosol. Spelling problem there, but still. This allows for a higher tolerance for higher salinity. Yes, that was, and there were others of you who also wrote very, very good answers for that. That's a good. Okay. All right. So that takes up a lot of our time, and I don't want to take up too much more time. Here's what I'm going to propose now. Excuse me. Okay. So Mr. Anderson has a really, really good presentation of this, and I'm just going to go in and let him talk to you. He does a good job. Second. Okay. That one. Okay, all right. I'm going to turn off my sound now so you can hear him.
Hi, it's Mr. Anderson, and this is a beginner's guide to graphing data. I continue to be surprised by the simple mistakes that are made by students in my high school science class who try to display their data in a graph. I think the reason why is that we spend a lot of time in elementary school on graphing, but as students move from middle school to high school and on to college, we don't spend very much time on graphing. Just like reading, we assume that they know how to do it. So I put this video together to show you the importance of graphs, the different types of graphs, and even give you some practice graphing some data. So first of all, let's start with the term graph. The term graph and the term chart can be used interchangeably. They mean essentially the same thing. And so you'll hear me go back and forth between chart, graph, plot. They're basically ways that we can take data and display it visually. So why do we use them? Well, let me give you an example. This right here is David Keeling. He spent years and years and years on top of Mauna Loa in Hawaii collecting data on the levels of carbon dioxide in the atmosphere. And let me show you less than 1% of the data that he collected. It's almost incomprehensible to us. When we get a huge list of numbers, we can't make sense of it and we can't find patterns. But if I were to show you all the data Mr. Keeling collected, we would find the Keeling curve. And this is one of the most famous graphs in all of science. It's basically showing increasing levels of carbon dioxide. You see annual cycling of carbon dioxide. This is very important because it's tied to global warming. And so when we can see the data, we can make sense of the data. And so there are 5 major types of graphs that I'm going to talk about in this video. They are line, scatter, bar, histogram, and pie graph. So I want to start with a little pre-quiz and see what you know. And so could you right now point to the line graph on the right? Let me show you if you're right. Line graph is going to be that one. Could you point to the scatter plot now? And what about a bar graph? Could you point to the bar graph? And the histogram? And hopefully you can point to then the pie graph. Okay, so how did you do? Did you get all those right? Hopefully. The ones that are confusing, obviously a line graph is going to be dots connected with a line. Even though we have a best fit line here, the scatter plot is not going to connect the dots at all. It's going to show correlation between number sets. Did you get screwed up on the bar graph? How do we tell the difference between a bar graph and a histogram? Bar graph, we're going to have space between the different columns. In a histogram, they're going to touch. And so the reason it's important that you know and can identify the different graph types is that as a scientist, you're going to collect data, and then you're going to have to choose what graph you're going to use. In other words, the data always comes first. Then you got to figure out what graph you're going to use. And so let's talk about 5 different types of graphs. First one is going to be a line graph. We're going to use a line graph if we're looking at change over time. And so in this one, we're looking at U.S. trade in goods and services. And so you can see that the dates are going to be along the x-axis, and the monies are going to be along the y-axis. So in science, maybe we're collecting data on an individual worm and how much oxygen it respires over time or consumes over time, then a line graph would be a great example. Let's say we're looking at a scatter plot. We're going to use that when we're correlating two number sets or two sets of data. So it's a correlation of variables. And so this scatter plot right here, they're looking at the eruption of Old Faithful, how long that eruption takes, and then the time between the eruptions. And you can see that there's a relationship between those two.
Okay, how are we doing, guys? Can you see why the graph is really better as a scatter plot than as a line graph? Can you see? I took off if you did a line graph. A line graph was fine for this one. I didn't take off for that. But now I think it's important that we differentiate. A scatter plot is great if you have two different variables and you want to see if there's a relationship between them. And that's really what those scientists were asking. They were saying, is there a correlation between salinity and how many of these muscles have this gene, right, for increasing the concentration of their cells? So that makes a scatter plot really probably the best thing to have used, even if you didn't use it. Now you know that would probably have been good. A line graph is more for situations where something is changing over time and it's only one thing changing, like one particular variable changing over time in a single situation, right? So I'm going to keep going.
Scatter plots are really important in a science classroom. If we're ever varying one variable, we call that the independent variable, and then measuring how that affects another variable, we call that the dependent variable, then a scatter plot is going to be the graph of choice. And so when you're doing that, make sure that you put the independent variable on the x-axis. So let's say that this plant right here, I'm doing an experiment where I vary the amount of fertilizer, and then I measure how much that plant grows over time. Since I'm varying the independent variable, I'm going to put that on the x, the amount of fertilizer, and then we put plant growth on the y. We look at a correlation between those two. So scatter plots are incredibly important in a science room. What about a bar graph? We use a bar graph if we're comparing two groups together or more than two groups. And so in this bar graph, we're looking at worldwide incarceration rates, and we're comparing different countries. Let's say in the lab, we were measuring how different colors of light affect the rate of photosynthesis. Then we could put the rate of photosynthesis on the y-axis, and then we'd represent each of those different colors of light using a different bar.
Yeah, again, a really good delineation here. So line graph is changing over time or over some very, very closely connected thing that's in one situation. A scatter plot is two different variables and you want to see if there's a relationship between them, like salinity and the appearance of a gene, or like, I don't know, I can't remember the one he used about the eruptions, something like that. And then a bar graph, the key word here is comparing groups, particularly if those groups down along the x-axis are not numerical, right? So different colors of light, and then the photosynthetic groups go on the y. That would be a good bar graph, right? Because colors of light are not numerical necessarily. Right? Or something happening in different beakers maybe that are not necessarily connected and with totally different chemicals in different beakers, and then you're looking for the response of a living cell to it or something like that. That would make a good bar graph where you're comparing different situations that you did in an experiment. I'm going to go past past a couple things here because I don't think you need pie charts. No. Yeah. Let's go past histograms. Yeah. Let's go here. Let's start with that.
Let's play with that for a second. Let's say I've got data from a pretend ice cream store that I have here in Bozeman. And on this data right here, I've got the average high temperature in Bozeman, and then I've got the number of ice cream cones that I was able to sell during these different months. So let's say I wanted to display, for example, all the different months and then the percentage of the whole of which I sold ice cream. I'd be looking at a pie chart. So I'd have all the different months as a different section of that whole pie. Let's say I want to just over time look at the average temperatures, the average high temperatures in Bozeman. What would I use? That'd be a line graph. Let's say I want to look at 3 specific months, 3 groups, and compare the ice cream sales in all months that start with the letter J. Then a bar graph would be great. And so let's do a real graph and show you all the elements of a graph. So let's say I want to look at the correlation of ice cream sales and average high temperatures at my ice cream sale or my ice cream store. And I would use a scatter plot. So let me show you all the important parts of a scatter plot. First thing you want to do is you want to figure out where your axes are going to be. So I'm going to put along the x-axis, I'm going to put the temperature, and then I'm going to put my ice cream sales on the y. Next thing I want to do is I want to have a title for each of those. So I'm going to put average high temperature along the bottom. Notice that I put in parentheses the units that I'm measuring it in. Then I'm going to put ice cream sales on the y-axis, and I'm going to put the units that I measure that on as well. What's next on a graph? The next is going to be the title. The title should tell you everything contained on the graph. In other words, I'm going to look at the correlation of ice cream sales and average high temperature. I'm going to tell you where it is, and I'm going to give you the date for that. Lots of times the titles are too simplistic. You'll have a title that's simply ice cream. You have to have way more information in the title. In fact, I should be able to look at the title, and it should be the story of everything contained within the graph.
Ding, 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 ding. See, I think so too. Not just me, not just crazy Mrs. Murdoch, okay? So the title really does need to be inclusive and descriptive, okay?
What's next? Well, on a scatter plot, we're going to have the data. And then finally, we should have a best fit line. Best fit line is going to be like an average of the data. If you're putting this in freehand, basically half of the dots on your scatter plot should be above it, and then half of them should be below. A good best fit line should never extend past your data. I shouldn't put like an arrow on the end of it, because really, I can only put a best fit line within my data. I can only interpolate within the graph. So these are all the elements of a graph. Good title, label the axes, make sure you have linear numbers. So you can see here that there's 15, 15, 15 between each of those major grid lines, and we're going to have the same thing here on the Y. You've got your data points, and then you have a best fit line. And so those are all the elements of a good graph. Another important thing that students tend to do is they want to include the number 0 here. You don't have to have a number 0. If your data doesn't have the 0 in it, then it shouldn't be on your graph. So let's look at a bad graph. Let's say I'm collecting data where I'm looking at the amount of fertilizer and how that affects plant growth. Let's say that a student gives you this graph. Can you find all the errors in the graph? I can find at least 10 probably. Number 1, let's start with the title. So it's got a title, but the title isn't very descriptive. The title should show me what's going to be on my X and what's going to be on my Y axis. So like relationship or the relation of fertilizer to plant height. Next thing, I've got plant height here, but since I'm varying the amount of fertilizer, my independent variable should be down here. So we would want fertilizer to be down here, and we want plant height to be on the side. We also want to make sure that we put units in parentheses that we measure it in. So we would have plant height over here, then fertilizer down here. This is a scatter plot, so you should never connect the dots in a scatter plot. Other things that we have is we have nonlinear scaling. So down here, you see it goes 5, 7, 11, 15, 17, 19. The distance between each of these should be equal. And then this number here, 12.5, doesn't tell me anything. I couldn't measure anything else on here because I'm just given one number. So you have to have at least 2 numbers on your grid line. You can also see that the best fit line is extending past the data. And so this is a bad scatter plot. So we're having quite a few errors on that. So you want to try to remedy that when you're making a scatter plot of your own. And so let's get to that. So basically, I've got 2 videos that I've made. One is on graphing data by.
All right, that's good. All right, so that was a lot. I gave you a lot today. I want you to go take a break. I'm going to make breakout rooms, and you now will have the rest of class to, if you want to read Z puzzles, you can. If you want to get ahead on the enzyme work, you can. It's already posted. The asynchronous work I gave Friday. Right now, that's all that's there. So let's just take a break away from your screen, and when you come back, you'll have the rest of class to catch up or go forward. Everybody good? Give me a thumbs up. You there? Yes? Okay, all right. Great, excellent. Okay. So definitely go away. Go away. No dancer. Put your feet in the sand, something like that. Rather.
Hello.
Oh, hello.
Can you hear
Hold
me?
on. Yes, I can. Hold on. Yes, I can hear you.
Okay, so do we just work on anything right now?
Right. So what you want to do, I think the first priority should be making sure those good puzzles have good grades for you, right? So if you go to remember I showed you where to go in Google. They're fresh up in your folder.
Are they the same like um that puzzles?
They're the same ed puzzles you did with me a couple weeks ago, where I was wearing an apron and I showed you the I showed you some dialysis tubing and some colors, right? And then the second one, I was working with potatoes.
Mm-hmm.
So they're both. I put them both in the same folder, redo folder, and I also gave you. I showed you your original grades in your Google Classroom, so you can go and check to see if you need to do them or not.
I don't see the grade in my Google Classroom.
Okay. Who I'm talking to, Mikhail?
Yeah.
Okay, I'll tell you what it is. Hold on.
Hooray.
Okay, so you got a 94 on the diffusion one, so that's good. You can leave that alone. But then you got a 60 on the potato one, so you may want to redo the potato one.
All right, let me redo the potato one.
Yeah,
Oh,
there you go.
well, thank you. Yep, and
You're welcome.
and after we're done with that, we just work on the enzyme lab.
Yeah, go get ahead on the enzyme work. Yeah, there's only two little things in there. There's one on Mr. Anderson explains enzymes, right?
Mhm.
There's the guided reading.
Are the ed puzzles from now on going to be all graded for accuracy?
No, I'm going to be very, very choosy about what I make summative. No, most puzzles are just going into practice. Okay.
Okay.
But every once in a while, I'm going to say, okay, guys, this one's going to be a lab summative one. Right? I'll tell you.
Okay. Thank you.
You're welcome.
Let me go to a breakout room.